And now I will tell you a few words about my thesis entitled Diversity Patterns in European Vegetation Linking Community Ecology and Macroecology. I will start with our motivation and a brief introduction of the topic of the thesis. I will start with the subtitle, Linking Community Ecology and Macroecology. Our vegetation science group in Masaryk University has a strong background and a long tradition in phytosociology, that is in the study of plant communities. And as Milan mentioned already, at the time when I started my PhD study, the huge database called European Vegetation Archive was just launched, thanks to Milan and many other people. So at that time, new perspectives, I would say, for studying plant community diversity over large spatial extent over whole Europe have appeared. In contrast, macroecology is quite a young field. It is interested in more general patterns and processes operating on broad spatial scales. And in 2012, Jan Beck and colleagues published this paper entitled What's on the Horizon for Macroecology. And they suggested several options how the field of macroecology could move forward. Among others, they suggested that new small grain, large extent data could be assembled, that macroecologists should consider local processes leading to larger scale patterns, and that the past should be explicitly included in analysis to understand the contemporary patterns. So we said, okay, we have great data, great opportunity to do such a kind of research, and at the same time new analytical techniques and tools such as expert systems for classification of vegetation plots or various modeling techniques appeared as well. So it was the right time, and we said, okay, the challenge is accepted, and we started this work. About the the first part of the title, Diversity Patterns in European Vegetation. I created this word cloud, which is based on the density of words used in my thesis. And it nicely illustrates what is my thesis about. The thesis doesn't have a single concise topic, I would say, and instead it is focused on various biogeographical and ecological aspects of species diversity of vascular plant communities across the European continent. With my colleagues, we were interested not only in patterns and describing them, but of course also in the underlying processes and explaining of the patterns. We focused mainly on diversity of local communities, but usually we did regional comparisons of this local fine-scale diversity. We studied several habitat types in the component studies, but mostly focused on forests and grasslands. And specifically, we studied species richness or alpha diversity, that is numbers of species co-occurring in plant communities, then landscape history effects on current species richness patterns, alien plant invasion, so not only richness of native species, but also of alien species. We were interested also in richness patterns of higher taxonomic ranks, namely in plant families. And finally, we studied decoupled or not correlated patterns of phylogenetic and functional diversities. The general aims of my thesis were to describe and map fine-scale diversity patterns in the European vegetation and to explain and hopefully understand the mechanisms and processes behind them. So my thesis consists of 5 studies. 4 of them are already published papers and 1 submitted manuscript. And I'm the first author of 3 of the studies. The first study is about alpha diversity or local species richness, if you want, of vascular plant communities in of forest vascular plant communities across Europe. Because the vegetation plots from the European Vegetation Archive do not cover the European continent entirely, there are many gaps. We decided to create first models and then predictive maps of alpha diversity of forest communities, which would cover also these gaps. We are also interested in defining species richness hotspots in forests, in factors determining the observed patterns, and we had some not very specific hypotheses. We expected some similarity in the patterns found in forest alpha diversity with overall gamma diversity. Several such maps were published in earlier studies. And mainly we expected differences in the patterns found among the broadly defined forest habitat types, namely deciduous broadleaf forests, coniferous forests, and sclerophyllous forests. So we obtained vegetation plots from the European Vegetation Archive, and these plots were classified using the expert system into the UNIS forest habitats. We performed some data cleaning. For example, we removed non-vascular plants from the plots. We removed very old plots. Some of them were not precisely localized. And finally, we obtained some 70,000 plots for our analysis. To build the models, we used predictors related to topography, substrate, climate, also to glacial history and paleoclimate, and to landscape or land cover in the surroundings. And within the models, we also controlled for varying plot size, for the effect of varying plot size. So we used this data, vegetation plots and predictors in a form of GIS layers and combined them. This was an input for random forest models, which is a machine learning algorithm combining estimates from a large number of regression trees. So finally, we created predictive maps of alpha diversity based on these models. Here are some results. Our models explain from 51 to 71% of variation in alpha diversity. You can see some additional statistics here, and no. Sorry, I'll use this pointer. Here you can see a map of alpha diversity of all forests together. So we found this gradient, let's say, from species poor northwestern Europe to relatively species-rich southeastern Europe. Sorry. And here are some older maps depicting gamma diversity, and if you compare the patterns on the maps, the scales are different, but the patterns can be roughly compared. So you can see that there are some similarities, for example, here in mountain areas of southern Europe, but there are also quite some differences. Here are other maps for the individual forest types. For example, you can see that in coniferous forests, species richness is more concentrated to mountainous areas and to calcareous bedrocks, while in deciduous broadleaf forests it's more scattered, and we found also some, let's say, unexpected areas of high species richness, namely in Eastern Europe, for example, this area of hemiboreal forests in Lithuania, or the oak forests at foothills of Romanian Carpathians. The predictors used for models, here are their contributions. Predictors related to the current environment in the climate, topography, and geology were the most important in the models, but also the predictors related to land cover and historical factors, for example, precipitation change since last glacial maximum in coniferous forests, or forest area in deciduous broadleaf forests, were also very important. Here I will very briefly show you the effects of the three top-ranked predictors for each forest types. So in case of deciduous broadleaf forests, the alpha diversity was best explained with temperature, seasonality, annual precipitation, and forest area. Here you can see the hotspots which we identified. I will move to coniferous forests. So areas with highest species richness in coniferous forests is related to limestone area in the surroundings of the communities. Species-rich forests are especially in mountains, so in topographically heterogeneous areas. And in case of sclerophyllous forests, we found hotspots in each of the three southern European peninsulas, and highest richness in these forests is related to pronounced Mediterranean climate with low precipitation throughout the year and high energy inputs. The second study was focused on effects of Holocene history on current species richness. It was led by Jan Divíšek and Michal Hájek. So Holocene history, compared with other historical factors such as glacial refugia, is quite omitted in studies of species richness. So we decided to fill this gap. We were interested if historical factors derived from fossil pollen data could explain the patterns of current species richness of these three broadly defined habitats: light forests, steppe grasslands, and dark coniferous forests in Eastern Central Europe. And for each of these habitats, we had a specific hypothesis. For light forests, we expected that the high current species richness may be related to the occurrence of temperate forest herb species in late glacial and the Holocene onset. because this species might have their refugia in this habitat. For steppe grasslands, we expected that their current richness may be explained by the occurrence of steppe species and by landscape openness during the mid-Holocene forest expansion. And for dark coniferous forests, we expected that the occurrence of spruce and fir forests during the late Holocene beach expansion may be the important factor. So again, we obtained vegetation plots from European Vegetation Archive. We performed some data cleaning, and this time we considered not only the total species richness, but also the richness of habitat specialists. We employed environmental predictors, again related to topography, climate, soils, and land cover. And regarding historical predictors, we followed up this paper by Jamrichova and colleagues, and they performed this principal coordinates analysis of pollen types from 112 sites for each of the time period that we consider. And we used pollen site scores from this first PCOA axis and interpolated them to have this kind of continuous surfaces with these predictors, and these scores on the first PCOA axis for each of the periods perfectly fitted our hypothesis. So in case of late glacial and Holocene onset, the high scores on the first axis corresponded with temperateness, or let's say higher occurrence of temperate forest species. In these periods, it corresponded with landscape openness and occurrence of steppe species, and here with a representation of taiga species. So we used boosted regression trees to analyze this data, and we built two models for each time period. One was based on environmental predictors only, and the other one on environmental and historical predictors combined. And we again created predictive maps of species richness patterns. The models explained from 60 to 72%. When the historical predictors were included, the explained variance increased only by 1 to 4%, which is not very much. But if we have a look here, these are the contributions of individual predictors to the models, and we can see here in the lower part of the table that if historical predictors were included, they had the highest contribution to the models in case of light forests and dark coniferous forests, and the third highest contribution in case of steppe grasslands. If we would have a look at specialized species richness, then here for steppe grasslands, the contribution of history is also higher. You can see here that these results confirmed our hypothesis, as temperateness is very important for light forests in late glacial and so on. Here are examples of the predictive maps. Here, the left side, these are maps based only on models where environmental predictors were included, and the right side is on models where both environmental and historical predictors were included. So we concluded that these maps based on both the groups of predictors are like better fit the empirical knowledge about the distribution of species richness. So definitely these predictors is worth to include. Yes. I think maybe I will skip this because in these historical predictors, we detected often nonlinear or complex effects on species richness. So here are some examples, but we can come to that maybe later if you're interested. About the third study, it is about alien plant invasions in European forests. And this study was led by Victoria Wagner. Alien species are maybe very problematic in terms of altering ecosystems or negative impacts on biodiversity in general. So there are some strategies of how to prevent and manage spreading of these species, especially when they become invasive. But what we are lacking in general are analysis of drivers of these plant invasions across different spatial scales. So our goal in this study was to understand possible drivers of alien plant invasions in European forest communities, and we built on this on the results of this earlier study led by Victoria, characterizing species pool of alien species in European forests. Again, vegetation plots obtained from European Vegetation Archive. For each species in the data, we assigned status native alien or of uncertain origin. We focused only on neophytes, not on archaeophytes, and we tried to eliminate for example, forest plantations were interested only in spontaneously established species. We performed stratified random resampling and obtained these approximately 16,000 plots. Here you can see their distribution in Europe. As a response variable, we considered these two metrics, relative alien species richness. That means the proportion of alien species within a community, and sum of alien species covers. As predictors, we considered again elevation, environmental factors like climate, soil properties, and we also tried to carefully prepare proxy variables of degree of human disturbance or propagal pressure. For example, I don't know if it is visible. For example, we used this matrix like human population density or forest fragmentation in the landscape or distance from the transport infrastructure and similar variables. And we also checked habitat type and region. We used generalized additive models and quantile generalized additive models to fit bivariate relationships between the predictors and the two response variables. We chose these quantile GANs because our data were zero inflated. Alien species were present only in, I think, 17% of plots. So we hypothesized that the response close to the maximum quantiles could be more informative, and that we can better maybe detect the trends using this quantile GANs. We used 90% quantile and 99% quantile. And then we checked predictor variable importance using random forests. These are results for the GANs. these are group estimates for individual forest habitat types from the UNIS. These triangles are the estimates, standard GAMs, and these these quantiles close to the maximum response. So depending on which metric here, which model we will follow, we can see that the most invaded forest communities are these temperate and boreal mountain Betulapopulus tremula woodland or Acidophilus quercus woodland. If we consider these estimates, if we go for more conservative estimates, then this softwood riparian woodland would be the most invaded, and also if we look at the sum of alien species covers, so the riparian woodlands are the hotspots of local invasion in forests. Here are some selected bivariate relationships, and you can see that indeed if the quantile GAMs are used, then only the trends, some trends in the data appear. So, for example, the invasion level drops with increasing elevation. It increases with increasing imperviousness, which is the proportion of sealed soil in the surrounding of plots. The invasion level drops with increasing distance to transport infrastructure and so on. But the problem is that the peaks in these relationships are largely driven by these regional hotspots of alien species richness. We used these 250 by 250 kilometers windows, and these are again GAM estimates for these windows. And here we detected, let's say, regional hotspots of local alien species richness in forests. And unsurprisingly, it is in, for example, in Netherlands and Belgium, or France, also Czech Republic, or this part of Poland. And the values of environmental predictors that are responsible for the trends in bivariate relationships are correlated with these regional patterns. These are contributions of individual predictors. So you can see that the models explained 40% here and 21% in case of some of alien species covers, and that habitat type and this assignment to the regional window are the most important predictors that we detected. In contrast, climatic variables are very weak, except this annual precipitation sum here, or even the predictors related to human disturbance are quite low here, ranked low. So we concluded that there are really regional hotspots across Europe in local invasion of forests, and that there are also areas exhibiting invasion depth. That means that it's difficult to to analyze this data, because in some areas the pool of alien species is apparently not saturated yet, and these hotspots are really the primary pattern in the data. The study number 4 is about species richness of plant families in European vegetation. Our aim was to explore representation of vascular plant families in different type of vegetation across Europe. We analyzed this for forests, grasslands, scrub, and wetland vegetation. And our hypothesis about the patterns which we can find was that there would be differences among different vegetation types, and that families would have higher representation in areas and conditions that are similar to those under which their ancestors evolved and persist. So this niche conservatism hypothesis. In this paper, we prepared many maps and basic overview, some statistics, and assessment of the representation of plant families in different vegetation types. And we believe that this data could be used as a potential framework or inspiration for further macroecological and biogeographical research. Again, we classified using expert system the data from European Vegetation Archive. The huge task here was to unify the nomenclature, which was largely done by Irena Axmanova. We followed the Europlasmed plant-based concept, and regarding families, we followed the World Plants database concept. We removed alien species of extra-European origin from the data because we were interested in native flora. And we calculated relative species richness of plant family in a plot. It's again a proportion from the total species richness. And we averaged this relative species richness from plots across the grid cell of 50 by 50 kilometers across Europe, and these average relative species richness were then mapped. Here are some examples. In the data, we had almost 11,000 species from 158 families. And in this table, there are 20 most frequent plant families in European vegetation. Here you can see the 20 most species rich families. They are largely the same as the most frequent ones. We can see here that some families like Asteraceae are confined more to open habitats. For example, in Rosaceae, there are many woody species in Europe, so they are better represented in forests or scrub vegetation, or Cyperaceae largely diversified in wetlands, so they are very frequent there. And here are some examples of the maps of relative species richness of families for Poaceae and Asteraceae. Poaceae are an example of a family exhibiting a relatively consistent pattern of relative species richness across different vegetation types. We can see that highest representation is highest relative representation is in northwestern Europe, and we hypothesized that this could be due to dispersal lack after the recolonization after the last glacial. So in these areas there are many more grassland species and unproportionally less species from families of forbs. So therefore the pattern looks like this. The other explanation may be that, for example, grasslands in these areas have been very intensively used for agriculture. So this may have caused impoverishment of their flora. But given that the pattern is also in forests and scrub, so maybe the let's say explanation based on this biogeographical history may be possible. Asteraceae, in contrast, this is an example of family exhibiting rather inconsistent pattern. We can see that in forests, high relative species richness is in mountains, while in grasslands they are best represented in the steppe zone. In scrub, it's rather scattered in southern and southeastern Europe. Asteraceae diversified since the Oligocene, and they have spread to newly risen open habitats. They are adapted to wind dispersal, so these patterns nicely fit these characteristics of Asteraceae, as they occur mainly in open habitats, open mountain forests, and so on. And the last study of my thesis dealt with decoupled phylogenetic and functional diversity of European grasslands. Phylogenetic and functional diversities, they are metrics that characterize evolutionary relatedness of species in a community and their functional characteristics. And these metrics may be very informative regarding the filters of coexisting species regarding community assembly mechanisms. Let's say prevailing or traditional perspective on the relationship between these two diversity measures, between phylogenetic diversity and functional diversity, is this trend that these metrics are correlated, and it means that the communities should exhibit either phylogenetic overdispersion and functional overdispersion. This situation is in theory explained by a competitive exclusion of similar species, or it should exhibit this situation. That means phylogenetic clustering. So the closely related species are also functionally clustered, so they are very similar to each other. And this situation is traditionally hypothesized that can be under strong environmental filters. That means there is some stress factor selecting similar functional traits of species. On the other hand, there are these situations in which we were interested in this study. These are called decoupled relationship of phylogenetic and functional diversity. So the situation of decoupled phylogenetic diversity means that there are trait convergence in distant lineages caused by some environmental filters. And number 4, decoupled functional diversity. It means that there is trait divergence in communities in close relatives. Okay, so we wanted to analyze or to explore the situations of decoupled diversity patterns, because it's largely understudied. So we chose European grasslands as model ecosystem. European grasslands cover a wide range of environmental conditions. They have very diverse glacial and interglacial histories. There are different types. So we decided that European grasslands could be a nice model system. Yeah. We have this specific hypothesis. We expected that the traditionally assumed coupled PDFD pattern would be the prevailing case, because it is related to long-term, relatively environmentally stable conditions, while we hypothesized that these cases of decoupled phylogenetic and functional diversity could be related to environmentally change-driven conditions, namely accumulation of lineages, for example in refugia, and coupled with long-term effects of specific environmental conditions causing their functional similarity. In this case, we hypothesized that this pattern might emerge from environmentally change-driven extinctions. It means that during the recolonization process, there may be a limited number of lineages available for this recolonization, and that species are filling free niches, and that there is a selection of not that similar species from the limited pool. Okay. So we again obtained vegetation plots from the European Vegetation Archive. We distinguished these broad habitat groups of grasslands: dry grasslands, mesic, wet, and alpine. We performed the data cleaning. We resampled the data in 2 steps. We tried to resample locally overrepresented areas as well as overrepresented regions. We considered angiosperm species only, unified the nomenclature according to the plant list, and importantly, we used only plots where at least 80% of species had available data on functional traits. So finally, we had this data set of 81,000 plots. Then we used a phylogenetic tree and data on functional traits from TRIDE database and CLOPLA. Specifically data on plant height, leaf area, specific leaf area, leaf nitrogen content, seed mass, and lateral spreading distance in clonal species. And we calculated phylogenetic diversity and functional diversity of individual traits as mean pairwise distance, and we calculated standardized effect sizes of this matrix by using 100 permutations and keeping numbers of species in a plot and randomly selecting species from the habitat group species pool. We quantified the degree of decoupling as orthogonal distance from the PDFD theoretical trend. So this trend corresponds with the theoretical expectation of the coupled PDFD, and we are interested in the distances of plots defined by this space of phylogenetic and functional diversity. So we are interested in the distances from the trend. These dashed lines corresponds to distances 1 and 2 in the standardized effect size units. So the farther from the trend the communities were situated below the line, the higher the tendency towards decoupled phylogenetic diversity, while in this direction there were higher tendency towards decoupled functional diversity. We used chi-square test to compare the proportions of plots assigned to this interval from A to F, and in case that the plots were significantly more represented in the intervals on just one side of the trend, then we suggested to call these communities having a high tendency towards decoupled diversity. Additionally, we used data from the CHELSA database and NVIREM, climate variables and topography variables, and correlated them with this degree of decoupling. We assigned the plots to this UTM grid cells, 50 by 50 kilometers, and calculated average degree of decoupling. Here is one example of the results. You can see here x-axis phylogenetic diversity, y-axis functional diversity of specific traits. So in this case, it's plant height and leaf area. These lines corresponds to the lines here. So the solid line represents the theoretical trend, and the dashed lines are the distances from the trend, and the colors indicate, in case of yellow colors of the plots in this space, these plots are very situated very close to the theoretical trend line, so they can be considered displaying a coupled PDFD pattern, while the red colored plots, the more red, the higher the tendency towards the decoupled phylogenetic diversity, and the more blue, the higher the tendency towards decoupled functional diversity. The violet lines are linear trends, like the actual linear trends from the data. So we can see here that there are no overall relationships, linear relationships between phylogenetic and functional diversities, but there are many communities exhibiting either the coupled or the decoupled patterns. This figure is maybe more intuitive. Here again, the solid green line represents the theoretical trend, and the dashed lines are the distances from the trend. And these beans represent the frequency of plots in the space. So at the x-axis, this is the degree of decoupling, the distance from the theoretical trend. So if the distribution of the plots is significantly shifted, in this case of plant height towards decoupled phylogenetic diversity, then we inferred that plant height in dry grasslands displays overall a tendency towards highly decoupled phylogenetic diversity. In case of leaf area, there is no significant tendency towards the decoupled diversities, and in case of lateral spread, this was the only case where we detected very evident tendency towards decoupled functional diversity in mesic and wet grasslands. Here are some maps depicting the patterns of degree of decoupling. So in case of plant height and dry grasslands, we found that the communities tending to decoupled phylogenetic diversities are mostly concentrated in Western and especially in Central European mountain areas, like here, while in Eastern Europe, the communities exhibit mostly either coupled pattern or slight tendency to decoupled functional diversity. We explain this pattern in a way that dry grasslands in this part of Europe are in much more, let's say, diverse environmental conditions. There were many shifts during transitions between glacials and interglacials, so the ranges of species and communities changed, and this might have allowed a mixing of species pools. And these areas are also climatically more mesic, so the grasslands are often semi-dry and contain mesic species, which again enlarge their phylogenetic diversity. So the phylogenetic diversity is higher in this area, and but still these are dry grasslands, so there is a stress caused by, for example, droughts and shallow soils. So the functional traits like plant height are rather clustered in these communities compared with these. In case of lateral spread, this is an analogy of the pattern that I showed for Poaceae when I was speaking about the relative species richness of plant families. So in these areas of northwestern Europe, there are communities dominated by grasses. So the coexistence of different species of grasses or graminoids in general is likely enabled by differences in their lateral spreading, in their clonality. We don't have very many data for this combination. It would be interesting to see whether the pattern holds even in other parts of Europe, but in Central Europe the situation is different, and the communities tend more to coupled pattern of phylogenetic and functional diversity. And in case of alpine grasslands, this example illustrates that these grasslands largely tend to decoupled phylogenetic diversity. Here I forgot to mention these are correlations with the environmental factors. The plots with higher tendency towards decoupled phylogenetic diversity are characterized or occur in areas with higher climatic moisture and with higher terrain roughness, so topographical heterogeneity. And it's possible that it's these areas where many lineages of species occurring in alpine grasslands might have again survived periods of climatic deterioration. So nowadays they have higher phylogenetic diversity, and alpine grasslands are typical of many stressors. There's cold or effects of wind desiccation. So these environmental factors work as filters and filter similar species, for example, in this case, similar in plant height or also similar in leaf traits. So the patterns looks like this. Here are some conclusions from my thesis. So although the general patterns which we found in local plant community diversity are often very similar to the patterns of regional diversity across Europe, we revealed quite some habitat-specific patterns. We found that the processes linked via species pool effect with regional history are very prominent drivers of local plant community diversity. We noticed that high local diversity is quite often related to a higher environmental heterogeneity and particularly to transitions between different ecological conditions, which is the case, for example, of those dry grasslands, which have the highest diversity in rather semi-dry conditions, and we found a strong phylogenetic signal in the richness patterns of plant families across Europe. And also we found these contrasting patterns of phylogenetic and functional diversities, these situations when the diversity is decoupled. So we think that this calls for further integration of taxonomic, phylogenetic and functional approaches in future research. So I'd like to thank you. I would like to thank all my colleagues with whom I could collaborate on these studies, and thank you for your attention.